I will shortly present guidelines for conducting genetic monitoring of Populus nigra in the field. European black poplar is a typical pioneer tree species with sporadically occurrences as many individual trees of group of over mature trees in mixed riparian stands. It is fast growing, dioecious species with efficient dissemination of seeds and good ability for vegetative propagation. Mature and offspring populations are not overlapping. Natural generation might be possible where fresh, weedless and grassless alluvial or ruderal soil is available. Successful natural generation is usually patchy and sporadic, but in many years also absent in riparian ecosystems. Populus nigra naturally forms metapopulation of interlinked local populations, rather than small isolated populations. Due to changes in site conditions, its population size may expand or contract over time. The main obstacle for phylogenetic monitoring is to find habitats where the mating system can develop in a natural way and offspring can find regular, suitable mid to long-term conditions. To ensure representative sampling across the metapopulation, it's important to design a phylogenetic monitoring system with randomly selected monitoring trees in local populations along the river system and randomly selected monitoring subplots with germinated plants in natural generation microsites. Forest genetic monitoring plot should be selected in riparian forests on alluvial sites where local population of Populus nigra are parts of the metapopulation. Information could be obtained from vegetation maps and forest management plans. You have to consultate with local foresters and join initial field surveys. Field surveys should be repeated also in early spring when the trees are flowering to ensure equal male and female sex ratio among selected trees. Because Populus nigra metapopulation is mainly exposed to high hybridization pressure from non-native Populus species and hybrids planted in production plantations, it is necessary to confirm taxonomic status of selected trees also by species-specific genetic markers. For a genetic area with 50 reproducing and pure Populus nigra trees of different genotypes should be selected in local populations in sex ratio 1:1 of male and female. The monitoring plot in each local population should include at least 20 adult trees in maximum distance of 5 kilometers. All 50 selected trees must be labeled with continuous numbers. Sampling design of natural generation follows the metapopulation concept of multiple natural generation centers to capture the whole genetic diversity of Populus nigra and to assess the risk of gene integration and hybridization from exotic poplar species in the given area. The maximum distance should be 500 meters in a radius of local populations where fresh alluvial could be deposited to ensure seedlings establishment within pollen and seed dispersal distances of all potential parents. The possible microsites of natural regenerations should be frequently monitored from April to June, and where germination plots are occurring, the seedlings should be sampled immediately. 100 newly germinated seedlings, ideally from 20 natural generation subplots of 1 square meter, distributed all over the given area, should be sampled for hybridization assessment and for selection of 50 pure Populus nigra plants for further genetic analysis. If this is not possible, 2 sampling alternatives are proposed, depending to the time frame duration with no detection of germination plots. Genetic tests on natural generation should be performed by diagnostic genetic markers for selections and for further genetic analysis of pure Populus nigra samples. The Populus nigra guidelines also provide information on recording all field level verifiers and phenotypic data at the basic, standard, and the advanced monitoring levels. Thank you for your attention.